that seems to be susceptible in autism, as well as various proteins around the synapse, which we then talk about: neuroligands, neurexins, SHANK3, PSD95. These are the proteins that seems to be involved. So these findings point to a core deficit in synaptic function in many of the genetic forms of ASD. So that is a critical point in order to identify, a potential treatment. And as I said earlier, what is important for us as EFPIA members is that we understand, and this is really a critical point, that we understand the underlying neurobiology of autism at this point in time better than we do for schizophrenia or for depression. That's very important. That's then also so an additional lever to that is the second key development, which is that we realize now that there's possible treatment for these disorders, and this is derived from, let's say, some monogenetic diseases and animal models, where it has been shown that key behavioral and neuroanatomical phenotypes have shown to be responsive to drug intervention. And obviously the leading concept in this field is from mGlu5 receptor antagonists for Fragile X, and that's how I got into this field. But there are other forms that there's very nice and critical confirmation of drug intervention possibilities here. This is rapamycin for tuberous sclerosis, statins for neurofibromatosis type 1, and IGF for Rett syndrome. Now, these approaches are currently in translation to the clinic, and in fact, companies like Novartis and Roche are running clinical trials already in autism in Fragile X. And Novartis is most advanced in this area. They have launched a phase 3 trial now, global phase 3 trial in Fragile X. It's very important that we realize that these approaches are currently in translation in the clinic, but they also offer a perspective for the control of beyond Fragile X, and particularly on autism spectrum disorder. We hope that some of these models that we have, and Fragile X as a prototypical disease, has some merit to move to the next level, which would be autism spectrum disorder. What we have to realize that this concept, what we're doing now, was not believed possible, let's say 5 years ago. So that's how short this span is, and how this field has moved so quickly. Because the field has moved so quickly, we actually now need this public-private collaboration. Because we, as EFPIA members, realize that we cannot change, Roche cannot change Europe in developing a concept to develop a drug in autism spectrum disorder. We need all of us, of the pharmaceutical companies, and you. And that's why we developed this call. So, major advances in the understanding of the neurobiology of autism spectrum, and there's this emerging awareness that it's a viable treatable condition now. This has to now translate into registered drugs. But at the same time, having all these advances, we also realize that there's no integrated research and development concept, if you will, in Europe to do that. If we compare that to the US, it's a totally different situation. There has been a remarkable effort of advocacy groups, and we haven't seen that really in Europe to come together actually to a field of drug discovery. That has developed in the US much better, I think. Now, to produce a real impact on this field, a united effort of various stakeholders, and these include academia, industry, regulators, foundations, and patient groups, is urgently needed. Because we are advancing rapidly, we have questions on how we want to develop our drugs, and we now need you in order to come together and provide a potential treatment for these patients, because we have together to pull this to a concept in order to get these drugs on the market and hopefully then treat patients. So what is what are the objectives of the full projects? Well, as I said earlier, as Europe has not very strong outline structures for autism research, I think we have also a very unique situation that everything we will do will have an impact, and we can actually now shape it together, not just EFPIA, but we together, academia, regulators, and patient groups, can create this context for drug development in a way we want it. And so create this European-wide strategy for ASD treatment, integrating public and private strengths. And this is really the strength here that we think we have. It's a public and private enterprise. So what we want to do, and this is then very important information for you, is to set new standards in research and clinical development to aid the drug discovery process, develop and validate translational approaches for the advancement of novel therapies to treat autism spectrum disorder, identify, standardize, and develop expert clinical sites across Europe to run clinical study and trials, and so create an interactive platform for ASD professionals and patients. And ultimately what we want to have, of course, is evidence-based treatment for ASD patients, which means the same diagnosis, the same clinical assessment, and the same outcome measures. I'd like to highlight, though, that what we're doing is not drug discovery. We're providing the context in order to allow drug discovery to do its work. That means all the research, drug development, etc. But we're not doing the drug discovery. That will then be done in the pharmaceutical companies. So what we do is everything that's in the context of pre-competitive nature. So connecting critical mass to exploit innovative advances in autism, standardization between public and private organizations, rapid sharing of information to ensure progress, and alignment across the whole value chain from research, clinical development, regulators, and patients. That's what we want to achieve. What will be the impact on the R&D process? Well, identification of reliable and predictable assays, both on cellular level, animal models, and translational models, that can be then also be transferred to various other laboratories. Standardization in research and clinical practice, alignment of the field, complete field, including regulatory praxis. And then obviously IMI is all about excellence of research in Europe. Ultimately, we want to develop Europe as an area of preference to run well-designed and controlled clinical studies with new medical entities for pharma and academia. How do we want to achieve that? Well, we have defined 6 work packages, and it's actually no rocket science. It's very, very straightforward. We have a work package on in vitro systems, in vivo systems, on translational and biomarker development, and on clinical research. And then we have 2 other work packages which I will also introduce. So the first work package is on in vitro systems, and this will take advantage of emerging advances in the field of autism genetics and aim to develop in vitro model systems for drug characterization and evaluation. So here examples are new assay development. This could be primary embryonic neuronal cultures, in vitro slice preparation. Think about the Mark Baer preparation of the hippocampal slice and LTD. Embryonic and induced pluripotent stem cells. I think that's a very interesting development that should be followed up. That could be done with patients' cells. And then define phenotype at synaptic and cellular and security level. It's all about characterization, where do we see change as opposed to wild type, etc. Determine then the reliability and reproducibility of assay. I think that's a very important part, because what we want in the end is that an assay that's being run in Helsinki has the same outcome as the assay that is being done in Rome. So this is really very much about reproducibility. And then use these assays to assess pharmacological tool compounds. And then ultimately, obviously, is the translation of assay endpoints to animal models and man. Work package 2 has the same outline, basically. Here we're talking about animal model development. And this work package should deliver recommendations on the most appropriate animal behavioral endpoints for the use of genetic and/or environmental models relevant to ASD. So this could be, for example, some transgenics or knockout animals. What we should agree on, and that's very important again for the alignment of the field, define translational measures, for example, on cognitive, affective, and social behavior in ASD animal models, develop and standardize the outcome measures of ASD animal models, and assess the reliability of reproducibility across collaborating labs. I think that's, again, a very critical step. I'm a behavior pharmacologist by training, and I know how difficult it is sometimes to confirm some of the animal behavior in different labs, even labs that are very close to each other. And obviously, the value of the animal models will be to assess pharmacological tool compounds that we at EFPIA might be providing then. Yeah, and obviously other relevant endpoints and recommendations. What I forgot to say in the beginning is that the outline of the call is obviously a recommendation. So it's also up to you as an applicant consortium to fill in the gaps or say, well, we give priority to some other packages or some other ideas. Work package 3 is on the translational research development. And this is obviously around developing objective biomarkers linked to ASD to translate, for example, electrophysiological imaging and pharmacological outcome measures from animal to man. But what we could also be doing is translating from man back to animal. So here we think that would be useful is to identify translational biomarkers of neuroanatomical changes in animal models, such as fMRI and PET and whatnot, or any other imaging approach that you might have. Define phenotypes in models, using markers to define shared common signatures among different ASD genes. I think this is a very important and critical part. I think if we could somehow identify common signature in the various models that we have based on the genetics and the different genetics that we have identified, a common signature would, for example, help to define us developers, drug developers, to identify relevant targets that are applicable then to a wider population. But also, these biomarkers would then help us, for example, to stratify clinical studies later. Then use these promising models to assess efficacy of testing pharmacological tool compounds in these models, translate endpoint from animal study to ASD patients and back, and establish the susceptibility for pharmacological challenges to any of the measures above. Then we come to the clinical research development, work package 4. And I think this is a package that is critical for the development of our molecules. And I think there's enormous amount of work to do here in Europe. So this work package should facilitate and enhance scientific collaboration and exchange across Europe for ASD professionals, and these are not only research, but it's also the clinicians for both private and public sectors, and to significantly promote research and development of drugs for ASD. So what we want to do here is actually first assess standards of care in autism spectrum disorder of patients in Europe. There's relatively little information on how ASD patients are being treated in Europe.